I'm going to switch gears to introduce the woman of the hour, Keren Lasker from Scripps Research Institute. Awesome. Thanks, Keren. She received her PhD in computer science from a plethora of universities, really. Tel Aviv University. She worked through UCSF and did some time at the Max Planck in Munich. And the highlight, she says, is that she got to solve the 26S proteasome structure. Wow, that was tough to say, mouthful, I guess. Then she went on to Stanford University to work with Lucy Shapiro during her postdoc, where she really laid some foundations on showing the functional role of phase transitions. And most recently, Keren's been working on designing condensates, as many of you have probably seen on our bioRxiv preprint that came out earlier this year. So Keren's actually just getting her lab up and running this summer at Scripps, where she'll be spearheading research on the structure and dynamics of bacterial condensates with an eye toward engineering condensates for eukaryotic cells. And to do this, her lab uses really cool high resolution imaging techniques complemented with powerful computer modeling. And so we're all really excited to see what the future has in store for her in her lab. And so thanks, Keren, for joining us today. We're all super eager to hear about your work today on the bacterial and designer condensates. Please take it away.
Thank you. Hello, everyone, and thank you, Jill and DuPont, for this kind invitation. I'm super excited for the opportunity to present my work as part of the Kitchen Table Talks. And today, I'm going to tell you about cellular organization in bacteria, and in particular, about one biomolecular condensate that resides at the pole of the bacterium Colobacter crescentus, and how it can be used for synthetic applications in mammalian cells. So just to give you an orientation of what we're going to be doing today, I'm going to introduce cellular organization in bacteria. Then I'm going to introduce Colobacter crescentus and the polar organizing protein POP-Z. Then we're going to talk about POP-Z condensation and its molecular grammar. We're going to talk about the function of POP-Z in Colobacter. And finally, about how we can use POP-Z as a synthetic biology tool. So cells are organized across many length scales, from the organization of atoms into proteins, proteins into molecular machines, and the organization of these machines within distinct subcellular membrane-bound organelles. We now know that in addition to these membrane-encased organelles, cells harbor a variety of compartments that lack a casing. These protein and RNA condensates dynamically assemble and disassemble, and they selectively permit entry of enzymes and substrate to carry out specific cellular functions. But what about bacteria cells? The size of a bacteria cell is a third of a mitochondrion. Do they also use this intermediate organizational scale? So until not long ago, it was actually thought that a bacteria cell is simply a bag of enzymes. But with the advances in multiple imaging modalities and the ability to look at dynamics of individual molecules inside the cytosol, it became very clear that the cytosol of the bacteria cell is both highly crowded and highly organized. And I'm going to list 3 such examples of such structure, although many more have been discovered and characterized in detail. So first, with the aid of nucleotide-associated proteins, the bacterial chromosome hierarchically folds into a compact DNA while remaining accessible for protein complexes involved in replication, transcription, and DNA repair. Second, the tubulin-like protein FDSE assembles at mid-cell to initiate cell division. Additional essential proteins are then recruited to the division site and activate cell envelope constriction. And finally, chemoreceptor arrays allow bacteria to assess their chemical environment and bias their movement towards a more favorable conditions. And really exciting for us is that we're now starting to appreciate, really in the last few years, that in addition to these highly ordered structures that have been studied for a long time, The bacteria cytosol actually includes several biomolecular condensates akin to their eukaryotic counterpartners. These include, among others, RNA degradation bodies, RNA polymerase bodies, phosphatase granules, and carbon fixing bodies. My work is focused on aspects of cellular organization in the bacterial model system Colobacter crescentus. Colobacter crescentus is a freshwater bacteria, part of a highly abundant and diverse alpha proteobacteria class. Colobacter goes through asymmetric cell division. So what do I mean by that? One life form of a Colobacter cell is a motile flagellated swarmer cell that cannot replicate its chromosome and is in a G1 phase. The swarmer cell then differentiates into a stalk cell by shedding its flagella, attaching to a surface, and synthesizing the stalk at the same pole where the flagella once resided. At this point, the cell switches to its S phase and starts replicating its chromosome to grow and to synthesize a new flagellum at the cell pole opposite the stalk. This process takes approximately 90 minutes in minimal media. Once DNA replication is completed, the cell enters the G2 phase, and cytokinesis of the inner membrane, using those FTSE proteins that I mentioned, compartmentalize the replicated chromosome into nascent swarmer and stalk progeny, so, giving rise to two daughter cells, the swarmer cell and the stalk cell. The swarmer cell, again, will swim around and eventually differentiate, and the stalk cell will immediately go through another round of the cell cycle. So how is this, and this process happens every cell cycle. It's very predictable and beautiful to see. So how is this process being regulated, especially given that cytosolic protein diffuse an average on a rate of 0.5 micron square per second, and that Colobacter has no membrane-bound organelles, and that the cytosol of the cell is only 4 micron long prior to division. So, I mean, wouldn't all possible signals just be scrambled in the cytosol of the cell? So it turns out, actually, that through many years of work of many groups, we now know that Colobacter achieves this exquisite asymmetry by sequestering antagonistic signaling proteins to the two poles of the cell. And what happens is that signaling proteins that promote this stalk cell fate that I mentioned are recruited to the all pole, and signaling proteins that promote the swarmer cell fate are recruited to the new pole, such that upon division, the nascent swarmer cell inherits the signaling pathway that will promote the swarmer cell fate, while the mother cell inherits a signaling pathway that promotes the stalk cell fate. Now, this sequestration of at least 13 different types of signaling proteins is mediated by direct and also transient binding to a disordered protein POPZ, which is the star of the show today. And what's happening is that POPZ self-assembles at the poles of the cell. So what do we know about POPZ? POPZ was co-discovered in 2008 in the labs of Lucy Shapiro and the lab of Christine Jacob Wagner. It is a cytosolic protein, 177 amino acid long. It consists conserved helical and terminal region, which was later shown to be used for client binding, and a helical C-terminal region, as well as a disordered region that is enriched with acidic amino acids and prolines. What else do we know about POPZ? Loss of POPZ results in multiple problems with the cell in morphogenesis, polarity, and cell cycle control. The cell will survive, but grow very slow, would lose all of these asymmetry hallmarks such as the stalk and flagella, and essentially it's just a mess. Also, something that is very interesting for us now is the realization that POPZ is conserved within alpha proteobacteria, but is not found outside of alpha proteobacteria. And again, alpha is a very, very diverse set of bacteria living in all kinds of conditions, and all of them have this POP-Z protein encoded. And here what I'm showing to you is a snapshot of the alpha phylogeny. And so far, POP-Z from 4 species across different order of the phylogeny have been studied. And in all cases, POP-Z was shown to self-assemble, to form a microdomain, and to play a critical role in cytosol organization. So we really believe that we're hitting here on a very important function and a very important process in cellular organization of alpha. What we also know that in Colobacter and in some other bacteria species, POP-Z was shown to exclude ribosome and DNA from entering the microdomain. So when I was in Lucy's lab for my postdoc, we wanted to understand those driving forces for selective entry. So now studying POP-Z in Colobacter is very challenging. Why? The size of the POP-Z microdomain is 250 nanometer, which is within the diffraction limit of optical microscope. So can't really do much, we think, but to overcome this challenge, we decided to perform single molecule tracking experiments of cytosolic proteins inside and outside the microdomain. And to perform these experiments, we collaborated with a super talented PhD student at the time, Lexi von Diesmann, and she was a PhD student in the lab of W.E. Merner at Stanford. And what I'm showing you here are single POP-Z molecules colored by the density, with red indicating low density and white indicating high density. And to start, we decided we'll start with just a simple control, nothing really interesting, just to measure the baseline diffusion inside and outside the microdomain. And for this experiment, we localized a heterologous PIF protein fused to IFP, with the assumption that a protein like that is not coming from Colobacter would not bind any Colobacter proteins, and because of that, it would freely explore the microdomain. But to our surprise, when we localized individual PIF-EYFP proteins, they did not enter the microdomain. Now, that actually was pretty striking. For ribosomes not to enter the microdomain, sort of the mental model we always had in our head is that it might be that POP-Z creates some sort of a mesh and large obstacles like ribosomes cannot penetrate. But here we just have a very small protein and we're still unable to enter this microdomain. But it's not that all small proteins cannot enter. When we look at additional cytosolic proteins, specifically we looked at Colobacter native cytosolic protein, CHIP-T here in orange and CPRA in green, both fused to EYFP, they did enter the microdomain as indicated by this red rectangle, while PIF, EYFP, and EYFP alone did not enter the microdomain. So what is going on here? Right? What is the mechanism behind this entry into the microdomain? Very suspicious. So we thought, okay, maybe it's a matter of size, as I mentioned, because of the ribosomes, but actually we saw that size is not a determining factor because larger proteins actually enter, again, indicated by those red rectangles, and small do not. Similarly, POP-Z is very negatively charged with this acidic disordered region. So we thought that maybe entry might be mediated by electrostatic interactions. However, charge does not seem to be a determining factor as proteins that do enter are at two ends of the charge distribution. But what we did find consistent is that binding pathway to POP-Z, meaning that if the protein directly binds to POP-Z, which is CHIP-T, we shown through SPR, directly binds to POP-Z, or has a binding pathway to POP-Z, in this case, CPRA binds to CHIP-T that then binds to POP-Z, it will enter the microdomain. So we were thinking, can we actually force a protein into the microdomain by changing its binding affinity to POP-Z as a way to test this hypothesis? So to answer this question, what we've done is that we've used optical dimer eyelid. And in this system, the eyelid lab domain has a low binding affinity to SSPB. Those shown here as the bait and the prey. And upon blue light activation, the eyelid changes its conformation, exposing a peptide that binds the SSPB bait. So what we've done is that we fused fluorescent protein to this eyelid, and we fused SSPB to POP-Z. And we asked whether we can force fluorescent protein into POP-Z simply by shining blue light. And indeed, we observed exclusion of the fluorescent protein from the microdomain in the dark, and rapid recruitment into the microdomain upon blue light activation of the binding interaction. And we've also observed that the fluorescent protein left the pole at a time proportional to the half-life of this eyelid SSPB binding. So that leads us to a working model in which entry into the microdomain is mediated by either direct or indirect binding to POP-Z. And we are now starting to work to understand the molecular features that drive this process. We also think that there might be competition involved between different clients, that there might be limited places to bind on the POP-Z matrix. And we're happy to get feedback and ideas later in the discussion session. Okay, so going back to our scale diagram that I've shown you before, so far I've told you about POP-Z localization in the cell and its ability to selectively recruit proteins. Now we're going to think about POP-Z as a biomolecular condensate and about the molecular features that are driving its properties. So we're heading now to the third section of the talk. And I'm going to show you that POP-Z forms droplets in vitro, in Colobacter, and in human cells. So we'll start with the in vitro data. So in collaboration with Ashok Denis and his postdoc Daniel Skoll, we recently showed that POP-Z creates droplets in vitro, as can be seen by this nice DIC picture. What we've also seen is that when we're building a phase diagram of the POP-Z condensate in sodium phosphate buffer, we build the phase diagram as a function of POP-Z concentration and magnesium concentration. We find that POP-Z condensation in vitro requires the presence of either magnesium or calcium. And building on results that I'm going to show you later in the talk, we suspect that magnesium or calcium bind charged residues of the disordered region and modulate these POP-Z interactions. And just for reference, POP-Z concentration in the cell is around 4 micromolar. And a recent paper measured concentration of ions in gram-negative bacteria, where Colobacter is one of those. And they are in the range of 130 to 270 millimolar. So those two range combined are within the regime that we see nice droplets in vitro. Okay, but that was in vitro. What's happening actually in Colobacter? So we would have loved to investigate those material properties of POP-Z in Colobacter cell. However, again, POP-Z binds membrane proteins, accumulates at the poles of the cell, and creates this 250 nanometer microdomain. So it's very hard to do any kind of in vivo assay, really. But luckily for us, really just by accident, we found that in cells with deformed shape, we were able to observe POP-Z leaving the pole as one droplet as the shape of the cell deforms, and immediately adopting a spherical shape that continues to bounce around in the body of the deformed cell. And here I'm showing you just one movie of this event, and this movie is in loop and it's taken in 1 frame per minute. And what you can appreciate is that this polar POP-Z, as the cell deform, is just leaving the pole into this extended body of the cell, immediately adopting the spherical shape, and it's going to continue to bounce around. This suggests that POP-Z is a liquid-like microdomain adopting a spherical shape to minimize surface tension. Further, as Colobacter is quite small, we decided to use mammalian cells as a test tube to study robustly POP-Z material properties in vivo. For that, we started to collaborate with Stephen Boynams, who is a postdoc in the lab of Aaron Gittler at Stanford. And Stephen really is the most creative and talented scientist and a great collaborator, and I hope you'll invite him to give a talk here sometime. So when Stephen was expressing POP-Z in the U2OS cells, we saw that it forms droplets outside of the nucleus. So many droplets, but all of them outside of the nucleus. And further, that these droplets fuse and merge, suggesting that indeed POP-Z phase separates into these liquid-like droplets. And another property that was really cool is that those selective features that I described to you before in Colobacter were actually retained in the human cells. So first of all, POP-Z phase separates separately from stress granules. But if we were to express the cytosolic ChIP-T protein from Colobacter in U2OS cells, they are immediately recruited into the POP-Z droplets. And another good thing is that I'm going to show you very soon a series of mutations, and all of the material properties that we measured for those series of mutation in Colobacter actually matched what we've seen in the U2OS cells, meaning that those are really are intrinsic properties to the protein and can really be used as an engineering tool. Okay, so I'm hoping that by now we are convinced that POP-Z creates liquid-like condensates, and we next want to ask, what is the molecular grammar of POP-Z? Okay, so it was shown in the past that the helical C-terminal domain of POP-Z, which is shown here as 3 helices, but now I've done alpha fold on it. It may be 2 helices, but we don't know yet. Is the one that drives POP-Z oligomerization. And indeed, if you were to delete the C-terminal region of POP-Z, either in Colobacter or in mammalian cells, you lose POP-Z condensation. So great. Further, deletion of another the conserved N-terminal domain, does not affect POP-Z condensation, both in Colobacter and in U2OS cells. In Colobacter, it would affect the ability of clients to bind. This is not something we're going to cover today. However, we were very puzzled with what happened when we were deleting the disordered region. So although we didn't see an effect on polar localization in Colobacter, in U2OS cells, POP-Z looked more like beads on a string versus these large condensates we see in wild-type POP-Z. And that led us to think about and to try to characterize this disordered region more carefully. So if we started to look at the amino acid composition of the POP-Z IDR, and this very high percentage of prolines, around 25% of the linker is prolines, and 25% of the linker is negatively charged residues. This looked to us quite unique. And indeed, when we projected the sequence composition of POP-Z homologs, shown here in red, and the sequence composition of disordered region from the entire human proteome, shown here in gray, doing t-SNE analysis, what we found is that POP-Z creates a distinct cluster, indicating that this type of amino acid composition of the POP-Z IDR is rarely visited by human IDRs. So you might say, oh, well, this is just because it's a bacterial IDR. So maybe they all look like that. But now in blue, we're projecting all the disordered region from all of Caulobacter proteins, and we see that this nice separation that we had before is lost. And that POP-Z clusters away also from most Caulobacter IDRs, with a few notable exceptions, including, for example, the disordered region of DNAA, which is which activates initiation in DNA replication in most bacteria. So there might be a really interesting thing here as well to look at DNAA sometime in the future. Okay, so our observation is that something is very unique with POP-Z IDR, and we decided to investigate the sequence of this, or the sequence features of this IDR further. And as we all know, it's very hard or almost impossible to do sequence alignment of disordered region because they don't align, because they're disordered. But what's very cool is that there are collective properties that are actually conserved. So what I'm going to show you now are 4 such collective properties. So the first one is the length of this disordered region. So in all or many of the POP-Z homologs in alpha proteobacteria, the length of the linker is conserved to be around 80 amino acids. And also, all of those homologs have around 25% of prolines in their sequence. Again, not alignable, but just collectively they have 25%. Further, they have 25% of negatively charged residues. And finally, really nicely, there is a nice mixing between those prolines and negatively charged residues along the sequence. And this mixing is conserved. So we're starting to think that, maybe these 4 properties are important for something. And when we started this project, we really didn't know what it's important for. So to understand why evolution maintained those 4 properties, we turn to do all atom simulation of the linker region with collaboration with Alex Olhaus. And what Alex simulation showed us is that these properties lead to a very high scanning exponent of 0.7, which means that it's a very extended linker and it's self-repulsive. So that led us to a model in which there is a balancing. I'm going to show you that in a few slides. That there is a balancing act between attraction forces implemented by this C-terminal oligomerization domain, shown here by those hands, and repulsion forces implemented by the linker. So to test this type of hypothesis, what we've done is that we've created mutants where we simply halved or doubled the length of the linker. And we found that dramatically affect the microdomain viscosity as measured by fluorescent recovery after photobleaching. And these experiments now are done in mammalian cells because it's much easier to get robust FRAP data. And what shown here is the fraction of recovery of the bleach spot after 60 seconds. In gray is the wild type POP-Z that shows a recovery of about 70%. But when we double the linker, the recovery is complete, suggesting a very liquid material. And when we halved the linker, the recovery reduced. So what was very nice is then we actually replaced the wild type POP-Z with either of those variants, and we wanted to see how is that affecting the viability of the cell using a spotting assay. And what we see is that in this double linker mutant, it's extremely toxic, and the cell is showing little to no growth, while having the linker shows slightly slower growth than the wild type. So what is going on here? So let's look at the localization of these linkers in Caulobacter. So here I'm showing you the 2 different mutants fused to M. cherry. And what you can notice that in the wild type and in the short linker, POP-Z is still residing at the poles. But in this super liquid double linker mutant, POP-Z is not maintained at the pole anymore, which is really the first time we see it. And I'm going to show you one movie that just shows you the extreme that this thing can go to. In this movie, this is an overexpression, but this liquid mutant is not just liquid within itself, but it's also just swimming around the cell, filling the entire cytosol of the cell. Now, you remember that I told you that ribosomes do not enter the microdomain. So what is happening in this liquid mutant? So to answer this question, we collaborated with Elizabeth Villa and her student Vincent Lam. And what they've done was acquire electron tomography of Colobacter cells. So they're freezing Colobacter cells, and then slice it with a focused ion beam and image a thin slice by cryo-electron tomography, allowing us to look at the finer details inside the cell in a very high resolution. So as a baseline, here is a picture of the wild-type POP-Z with a polar POP-Z that is residing at the pole and excluding ribosomes that I've told you before. Those ribosomes are segmented in gold. And also here you see a segmentation of the inner and outer membrane. But now, the picture to the right, we're actually able to capture this liquid POP-Z as it's just cruising across the cell, and we confirmed it with a correlative fluorescent imaging. And what is super cool is that now we have this liquid droplet that, again, is not maintained at the pole anymore. And it's just sliding around the cell, excluding ribosome and DNA. So what we believe that one of the reasons that this double linker is so toxic is that this complete loss is that changing the material properties of POP-Z leads to complete loss of cellular organization and constant reshuffling of the ribosomes and nuclears. Okay, so remember I told you that we have this model of attraction forces and repulsive forces. And what we've actually done by making the linker longer is essentially changing the stoichiometry between those attraction and repulsion forces. So what we wondered is, can we rescue this phenotype by correcting the stoichiometry? And to do that, what we've done is that we've created the wild-type oligoization domain is this trivalent 3 helices. We extended it by repeating helix 3 and 4. And what was very nice to see is that if we now use this double linker, but use this pentavalent oligomerization domain, we're actually able to go back to the material properties as we have in the wild type. And as a control, if we just use the pentavalent with the wild type linker, we're actually getting a pretty solid POP-Z microdomain. And also very nice is that if we're going back to our viability assay, we can see that restoring the stoichiometry actually restores the viability of the cell. So we actually continue to doing these type of experiments, because so far I've just talked to you about the length of the linker, but we actually also looked at the percentage of charge residues and prolines and their mixing between them and positioning and so on and so forth. This is in the bioarchive paper, you can see. But this slide really summarizes a lot of different mutants. And I think that this is really an important point. So on the x-axis is the calculated mobile fraction. So the more right you go, you're more liquid, and the left you go, you're more solid. And the y-axis tells you the fitness, and the higher the number, you're more fit. And what we see is that there is this type of Goldilocks effect where you have to be, if you're too liquid, the cells are not going to survive. And if you're too solid, also the cells are not going to survive, probably for a different reason. So the reason that we believe that the reason that evolution maintained this properties of the disordered region is really to tune the material properties to be just at the right spot to create those microdomains, which I'm going to tell you the additional function in a second, to create those microdomains that are required for cell organization, but also keep them intact at the poles of the cell, at the right size, and material properties. So to summarize this section, I hope that that you now agree with us that POPSI employs this modular design, and we are breaking it into three modules. There is the driver domain, which is this C-terminal domain, which is the organization domain. There is the disordered region, which tunes the material properties, and the N-terminal domain, which I haven't talked about a lot here, but there is really nice work from Grant Bowman's lab showing that the N-terminal domain is required for client binding. So that sort of, we see it as a domain that can give you function. And later, if we're thinking about designer condensate, of course, you can change this functional domain to be whatever you want, but you're still going to have those two modules of the driver for oligomerization and a tuner. Okay. So now I'm going to give you a little bit more information about what is POPSI actually doing in Caulobacter. And for that, we actually have to go back to this asymmetry story. And if you remember, I told you that the beautiful reason that POPSI maintains this asymmetry is that POPSI allow for sequestration of antagonistic signaling proteins to the two poles of the cell. But now let's go a little bit deeper in mechanism. So it turns out that what largely determines a swarmer sulfate versus a stock sulfate is the availability of a master transcription factor, CTRA. And active CTRA, which means phosphorylated CTRA, would promote the swarmer sulfate and inhibit the stock sulfate. And what is happening is that those signaling proteins that I told you, those that are residing at the new pole, would actually lead to activation of CTRA. And those that are residing at the old pole will actually lead to deactivation of CTRA, such that once a cell divides, you have a swarmer cell that is full of active CTRA, which would promote a gene expression of genes that are needed for swarmer sulfate. And the stock cell will be cleared of CTRA, allowing initiation of DNA replication. So the sole source of phosphate for CTRA is a membrane-bound autokinase called CCK. And CCK is one of those POPSI clients. And it binds to POPSI, and we've shown it. And it also acts as a kinase specifically at the new pole of the POPSI microdomain. Critically, CCK can also function as a phosphatase by a distinct biochemical mechanism. And CCK acts as a phosphatase everywhere else aside from the new pole. And the last layer is CHIP-T. So for CCK to pass its phosphate to CTRA, it goes to this intermediate cytosolic protein, CHIP-T. So when CCK acts as a kinase, it's autophosphorylating, passes phosphate to CHIP-T and then to CTRA. And the back transfer of phosphate is from CTRA to CHIP-T to CCK, permitting the dephosphorylation of the pathway. And then when we actually image those three proteins with respect to the POPSI microdomain, we saw that they're all enriched within the microdomain. So I'm going to give you two quick conclusions from our previous Nature Microbiology paper about the functional implication of sequestering this whole pathway inside a POPSI microdomain. And the first one is that POPSI defines a macrodomain for high kinase activity of CCK, reinforcing the asymmetry, and that localization of CCK pathway to the microdomain is required for generating a robust gradient of CTRA activity, which is needed for maintaining asymmetry. So I'm going to show you first why concentrating CCK inside POP-Z is important for its autokinase activity. So Tom Mann, which was at the time a PhD student in Lucy Shapiro's lab, created a synthetic liposome in vitro to test the effect of surface density of CCK, membrane-bound, autokinase activity. Now, it's important to note that CCK is a tetramer in vitro. And what he did, he loaded purified CCK onto the surface of this liposome via his anti-attachment, and he kept the total amount of CCK fixed, but spread them onto increasing amounts of liposomes, and measured the autophosphorylation of CCK through a radioactivated beam. And really, his beautiful result is that the autokinase activity of CCK, but actually, very importantly, not the phosphatase activity, is density dependent, meaning that for CCK to achieve high kinase activity, CCK molecules need to be clustered together. And POP-Z is the one that's going to help them do that. So when we went back to our single molecule experiments with Lexi, we constituted single CCK molecule localization for many cells. We observed that POP-Z defines clustered zone of CCK, meaning that CCK cluster is defined by where POP-Z is. It's not going to extend. And that the surface density of CCK is highest at the new pole microdomain. And when we are looking at, and what's really cool is that the concentration of CCK at the new POP-Z new pole microdomain is matching what we see in vitro as leading for, it's concentrated enough for autokinase activity. But everywhere else, the concentration is not sufficient for autokinase activity. So by having POP-Z allowing for CCK molecules at the new pole and maintaining POP-Z at a specific size, we can reinforce this autokinase activity at one position, patterning the autokinase activity of CCK. We also saw that now we move to do more detailed imaging for the dynamics of those three proteins with respect to POP-Z. We saw that although CCK molecules that are inside the POP-Z microdomain are mobile, they do have a reduced diffusion inside the microdomain, but they still move. And even more strikingly, the cytosolic protein CHIP-T and CTRA, upon entering the microdomain, they slow down by at least tenfold in their diffusion, leading to increased dwell time within the microdomain. They're still moving. They can still find their partners for those phosphotransfer, but the movement is slowed down. And we have this increased dwell time. So why is it important now to have those three proteins inside the microdomain with this increased dwell time? So to understand that, we've done reaction diffusion modeling, and we wanted to understand the benefits of sequestering this density-dependent kinase, CCK, and those substrate inside the microdomain. So based on published literature, we know that CCK kinase is mostly at the new pole, CHIP-T phosphorylation is also mostly at the new pole, and CTRA creates a gradient of phosphorylation. But what we've shown is that this polar sequestration, so now we tried modeling to see what would happen if we actually remove this polar sequestration of CHIP-T and CTRA to the microdomain. And what we see through our modeling is that if those secondary transfer from, if CHIP-T and CTRA are not going to be in the microdomain, we're actually going to completely lose the patterning of a CTRA gradient, which is again required for symmetry. And less profound, but still we're going to see this effect if just one or the other is going to be inside the microdomain. So to sort of test, to do some perturbation of this localization in cells. We overexpress POP-Z, and what it did, it led to enrichment of those three proteins at the old pole now versus the new pole. And what was interesting is that we then wanted to see if that would actually affect the ability to activate CTRA. So we've done qPCR of CTRA regulated genes, and we've seen that if we're actually changing now the relative sizes and composition of the POP-Z microdomain, and as an effect, the localization of this pathway to those two microdomains, we're actually reducing the expression of CTRA regulated genes, suggesting that indeed the composition of the POP-Z microdomain affects the patterning availability and activity of CTRA. And circling back to the previous part of the talk, when I was talking about material properties, we're now starting to connect material properties to this pathway. So what I'm showing you here is actually those 5 mutants that I told you about that have different material properties of POP-Z. And I'm showing you to the right now one of the expression of one of those genes, the proxy for CTRA activity. And I think it's very compelling to see that indeed we again see this relationship between this Goldilocks kind of effect, where the material properties would affect the amount of activation of this master transcription factor. So we think that, I mean, it's just the beginning. Of course, POP-Z has many functions and talks to so many proteins in the cell, but I showed you at least 2 ways in which the material properties affect cellular organization, both in the distribution of ribosomes and also in availability of active master transcription factor. So to summarize this section, what I'm hoping you're now convinced is that coordinated signaling within this POP-Z condensate facilitate asymmetry patterning of this master transcription factor CTRA, and specifically that entry into the POP-Z microdomain is selective and requires the binding pathway, and that selective sequestration into the POP-Z microdomain reinforces spatial regulation of asymmetry. And finally, for the last part of the talk, I want to give you sort of a primer of how we're thinking about POP-Z as a synthetic biology tool. So I told you that we have this modular view of the POP-Z protein with this driver, tuner, and actor. And for synthetic biology applications, we think that we can actually just now start playing with the actor to do different things. So this driver, which is essentially the C-terminal domain, we call it POP tag. The tuner is this disordered region that we now have a lot of information of how to connect sequence properties to the resulting material properties. And in an N-terminal domain, we can fuse novel functionalities. And here are a few more experiments that Stephen did. So first, I'm just going to show you that we can direct POP tag to different cellular addresses in the U2OS cells. So first, here I'm just showing you that the POP tag itself condenses in U2OS cells. You don't need the entire protein. I mean, you don't need the modulator and you don't need the actor. You just need the driver. But then you can start condensing different things. So here, for example, the POP tag is recruited to actin filaments, and we start to see a condensation of those actin filaments. Similarly, here is recorded 2 microtubules, and we can start to see condensation of microtubules. And this is my personal favorite, is that if we tag it to lipid droplets, we can actually create POP-Z shells around lipid droplets. And of course, there are many other cellular addresses that we can use. This is just to give you a highlight of or a taste of what we can do. Okay, so far I've shown you that we can take that the POP tag would condense in U2OS cells, and that we can addressed, directed to different cellular addresses. But what about the composition? I mean, can we actually change the composition? And this is where the actor, the N-terminal domain is coming. So what we've done here is that we essentially fused a nanobody to the N-terminal region of the POP tag. And by doing that, we can now control droplet composition. So in the first experiment that Stephen did, he showed that we can actually use a GFP nanobody and recruit GFP proteins into POP-Z condensates. And just to show you that this is not just GFP, we can actually now use GFP. We can now recruit stress granule proteins, which before I showed you they phase separate separately from POP-Z. We can actually recruit stress granule protein inside POP-Z. And also very cool is that we can deplete the nucleus from proteins by asking a protein that's supposed to be in the nucleus to bind to POP-Z. POP-Z would be outside of the nucleus, but then we can actually push protein away from the nucleus, depleting it from this protein. So this is sort of the setup. We're now working on various applications to use the system for engineering, and we're always happy to talk about possible ways to use it. And we're now also working on seeing how changing the material properties, given everything we know about the POP-Z linker, can affect function in U2OS. So to summarize this talk, what I hope I've showed you is that POP-Z creates liquid-like droplet in colobacter cells with selective entry properties, that POP-Z architecture is conserved across alpha proteobacteria, that POP-Z can be engineered to accommodate a range of behaviors. POP-Z creates a zone of selective activation and asymmetry determining kinase, essentially driving, reinforcing asymmetry in colobacter. And the POP-Z can be engineered to drive phase separation in human cells. And acknowledgement, there are so many people I should say thank you to. First of all and foremost to my postdoc advisor, Lucy Shapiro, was very supportive throughout the entire postdoc process, taking a person that did a PhD in computer science and allowing her to start pipetting in the lab. Various people from my lab, especially Tom, Emma, Shofen, Daniel, to my awesome collaborators, Lexi, Stephen, Elizabeth, Alex, and most recently Ashok and Daniel, and for all the funding agencies along the way. And I also want to give a very, very special and warm thank you to the wonderful community of Condensates, and especially to my close friends at IDP Sync and IDP seminars who really made a huge, really were instrumental to my career. And I feel that being part of this community was just really a game changer for everything that I was thinking of doing. And we do have an early career virtual poster session coming up, so please check that out. And lastly, oh, they don't have the slides. Oh no. Oh, sorry. And lastly, I just started my lab at Scripps. If you're interested in this type of work that we are doing, please consider joining us. We already have 2 members in the lab, Sia and Asma, which are a research associate and a postdoc. And yeah, that's it. I'm happy for the discussion. Thank you.
Awesome. Thank you so much, Keren. That was a pretty awesome talk. I must say that my favorite was the video of the really liquid linker condensates. That's like magical looking. Our first question, Charlotte, are you still around? Do you want to ask your questions about the IDR and?
Yeah. Can you hear me okay?
Yep, you sound great.
Great, okay. Hi, so my first question, you showed, I was also struck by the video of the extended IDR. And I was wondering if under non-overexpression conditions, if it still is properly localized?
No. So this is the picture that I've shown before. It still creates those droplets outside of the pole.
So do you think if you were to, like analogous to how you doubled the amount of helices on the C-terminus, if you were to double the N-terminal portion, do you think you would recover localization?
Yeah, we've shown that. We can recover it.
Okay,
yeah.
and then my second question is sort of also relevant to the last portion of your talk with the more synthetic condensates. And I was wondering if the kinase activity of the phosphorylation cascade that you were showing, is that affected by the localization and properties of the POP-Z condensate? And then sort of in a synthetic context, do you think that you can then tune the length of the IDR to sort of modulate enzyme activity?
Yeah, so you're hitting exactly where we're going. And this is the reason I wanted to show this, because I think we now have a system which is not too complicated, but complicated enough to really start teasing out this relationship between phosphotransfer and material properties. I do think that there is that there is something there, because in mild mutation where we just have the linker to be half the size and it's not really dramatic, we can still see effect on in the end of the day activation of this mass transcription factor CTRA. So I think that there is something there, but we're really now, I think, can start and we'll figure these things out in more detail.
Thanks.
Yeah.
Very fair. I see we're at the top of the hour, so if anybody has to drop off, you're free to go, but everybody else stay along for the discussion. We have a question from Guanha. I'm sure I'm slaughtering your name, so I'm sorry. But if you'd like, welcome.
Yeah, you're pronouncing correctly. So yeah, I saw that you changed the length of the IDR to make it half or double, and obviously the double like messed up with the material property and also the growth. But I wonder how adding like 2 helix repeats like 3 and 4 could rescue this phenotype in terms of the growth. I just don't get the logic here.
So what happens is that this was Charlotte's question as well. I probably should have shown that in the slide, but once we are adding those 2 helices, what actually happens is that the localization is going back to be polar. So first of all, we are actually losing this liquidity. So there has to be some valency relationship here. And what's happening, what we think that is happening is that potentially extending the linker actually inhibit the ability of POP-Z to bind other POP-Z molecules. And by extending now this oligomerization domain, we're adjusting back the valency to allow for interaction between POP-Z to POP-Z molecule. So pieces of data that we now have are first that if we just by FRAP, we are recovering the material properties close to wild type just by extending that. Second is that in Colobacter, the localization of this pentameric double extended linker is back to like wild type. So between those, what we're actually now trying to do are more detailed experiments to really understand what is the relationship between the IDR and this organization domain. Is it that the IDR is masking out potential binding interfaces on the organization domain, or is it something else that is happening between IDR to IDR? So we have a series of experiments that we're gonna try to do in the future, but just by studying material properties, we can see that we are back to the viscosity of the wild type.
I see. Yeah, this is also, I think, puzzling because I saw like the IDR is not essential for oligomerization, not essential
Right.
for helix interaction, but somehow it messed up with the.
Yes. So, okay. So what we're thinking that is, we have other section of our work that I didn't present here today, but I've done a couple of simulations, and we can show that there is a competition between POP-Z to POP-Z interaction, inter versus intra IDR oligomerization domain. And you can essentially have the sorted region bind to the oligomerization domain, and at this point, you're going to lose valency to additional POP-Z molecules. And that would create, would essentially break your condensate. So we were able to show that when we were actually changing the distribution of the charge residues to be close to the oligomerization domain or further from oligomerization domain. I didn't cover these parts today, but those are sort of the clues that we have now, and we're now going to do some single molecule threat and other experiments to really tease out this relationship between intra versus inter POP-Z interactions.
I see. Thank you. Looking forward to for the updates. Thank you.
Thank you.
It looks like Yifan has a related question. I'm not sure if you answered it about Yifan, are you still around?
Yeah. Thanks for the talk. So I'm wondering, like, the POP-Z interaction, if it only depends on the helix interaction, then this is going to be a fixed stoichemical ratio for such interaction. So how can you differentiate co-localization with phase separation?
So first of all, regarding your comment on fixed, so what I didn't mention to you at talk today is that at the end of this C-terminal domain, there's actually a patch of positively charged residues that are highly conserved and are important for creating for additional valency. So we're not sure again what is the how many ways there is for inter-POP-Z interaction. And second, regarding the localization versus phase separation, I mean, yeah, that is a very important question. What actually makes POP-Z localized to the pole of the cell? And we do have some additional unpublished data that shows that it has to do with a lot of it has to do with other forces that are happening in the cell, such as the DNA and the material properties of POP-Z, and how those sort of opposing forces work together to localize POP-Z. So this is why we're using this mammalian cell system to show you that POP-Z does phase separate. In Colobacter, it also localizes to the pole for various reasons that are have to do with cellular organization. But when we're changing the material properties to for example, be too liquid, those considerations are just out the window, and POP-Z is just losing its localization abilities, even though it can still bind to all those membrane polar proteins.
Thank you.
So many variables, it sounds like to me. We have a really cool question from Shruti. Shruti, are you around about the flagella in stalk?
Yes.
We can hear you. Go ahead.
Yeah, the reason I was curious because these two cell types change when the flagella becomes a stalk, right? Like the flagella finds like a place to attach, and then it becomes a stalk, and all of a sudden it's a different cell altogether. So I was just thinking, where does this change like arise from? Could it be like, once the flagella is not really operating, because how flagella operate, it's the whole new story of its own, like it has its own ways of operating. Once that stops operating or it becomes a stalk, could it be that something happens to the POP-Z condensate that causes to promote different kind of protein content in that condensate? I was just thinking in that direction. Would that change?
Yeah, so in this differentiation from swarmer to stock, actually a lot of things are happening, and various clients are being destroyed and new clients are being synthesized and entered. So what's happening during this differentiation is actually also changing of the client composition within the POPC microdomain.
Mm-hmm. Okay.
Cool. Awesome. So Talita, it looks like we have the last question. Do you want to unmute and ask?
Yes, I was wondering if you know if the IDR portion of POPZ expressed alone allows the bacteria to survive, or have you tried to express this IDR alone to see what happens in the bacteria?
We haven't expressed just the IDR by itself. It's just going to be diffused based on first one we've done, but we haven't checked for what it does to viability. But it's not going to create any polar condensate.
Very good. Well, that was very enlightening, and I hope it was very inspiring for other people and their next experiments, because there are so many details that you covered, and we just really appreciate you sharing them all with us. So thank you so much, Keren. It's been really lovely to hear your talk. And I guess you're seeing rounds of applause from the globe. So we'll close it down, and thanks again for coming. See you all next time.